Hi there, welcome to the virtual Beetroot Lab. My name is Vincent, and I'm one of the teachers teaching the biology course. I will be guiding you through this virtual experiment with a series of short videos. Let's start with the purpose of this lab. In this experiment, we are trying to investigate the effect of temperature on membrane permeability using beetroot as our specimen. The scientific name of the beetroot that we are using is Beta vulgaris. Beta is the genus name encompassing all different types of beets, while vulgaris is the species name. Derived from Latin, vulgaris means common or ordinary. In essence, Beta vulgaris refers to common beet, the type that you will find in supermarkets. Before we start the lab, let's review what we already know about the cell membrane. In class, we learned about the fluid mosaic model that describes the cell membrane as a dynamic structure comprised of various molecules. The major component of the cell membrane is phospholipids, which are molecules that has a hydrophilic head, shown here by these green circles, as well as two hydrophobic tails, shown here in yellow. When you have two sheets of phospholipids arranged this way, you form the phospholipid bilayer of the cell membrane. The second key component of cell membrane are proteins. Proteins can be integral proteins, which spans the entire thickness of the cell membrane, or they could be peripheral proteins that just attach themselves either on the outside of the cell membrane or on the inside of the cell membrane. In addition to proteins and phospholipids, there are other molecules like cholesterol, which are important for maintaining the fluidity of the cell membrane. And there are glycoproteins and glycolipids that plays a role in cell-cell recognition, for example. What's fascinating is that these molecules are not rigid. They are not frozen in place. Instead, they are free to move past each other and create this dynamic fabric that forms the cell membrane. So why do we choose beetroot as our specimen? Well, actually, there are several reasons why we do that. First of all, beetroot has a very vibrant color, the red color, right? It's very distinctive. And that color is actually as a result of a combination of several pigments. But the main pigment that accounts for this bright red color is called betanin. What makes betanin particularly suitable for our purpose is that it is water soluble. That means it can dissolve in water and we can observe it easily. More importantly, this pigment is uniformly distributed throughout the beetroot. That means you would expect to find similar amount of pigment in the core of the beetroot or on the edge of the beetroot. It doesn't really matter where you cut the beetroot from. You would expect to find similar concentration of the pigment. Now, that's not necessarily true for all plants. If you think about tomatoes, right? The tomatoes is red, it has its own pigment, but the color is not uniformly distributed throughout the fruit. Closer to the center, the tomato might be a little bit more pale, and you have some seeds that are kind of going into your way, and the fruit it's just not as uniform as the case for beetroot, and there's just less consistency associated with that. Lastly, you can see that the structure of betaline here on the right hand side is actually very, very big. And big molecules don't cross the cell membrane on their own. And that is important. That means when the cell membrane is intact, when the integrity of the cell membrane is not compromised, the pigment should stay within the cell. Right? So when we do our experiment and we see that the pigment is leaking out into the solution, it's dissolving in the water, that is an indication that the cell membrane structure has been altered or it has been damaged. So what kind of factors could potentially affect the membrane permeability? Well, the membrane permeability is related to the structural integrity. So anything that affects the structure of the cell membrane is potentially going to change the membrane permeability. Take temperature, for example. We learned in chemistry class, for example, when you apply more heat to molecules, they tend to move faster. Right? So you think of all these molecules in the cell membrane. If you crank up the temperature, they too probably will be moving faster past each other, right? And as molecules go faster and faster, they might leave more gaps, more space between each other. And that could cause the cell membrane to have more holes in it and therefore become more permeable, right? So is that actually the case? Well, we don't really know until we do the experiment, okay? But certainly, um, that's one theory. Type of solvent is probably going to affect the permeability as well. Remember, in living organisms, the environment of the cell is mostly aqueous. Okay, it's just water, right, in the surrounding, and that is a polar solvent, right? Water is polar because of uneven distribution of electron. But what if you change that to like a hydrophobic environment, right? That could cause the structure of the membrane to be completely different. Remember, the uh, right now the hydrophilic heads are all facing outside of the cell as well as the inside of the cell because those are aqueous environment. But if the environment becomes hydrophobic, then all of a sudden those hydrophilic heads want to get as far away as possible from the hydrophobic environment, right? So you might cause the membrane to flip completely, right? And that is certainly going to have an effect on the membrane permeability. pH is another factor. Remember, you have all these proteins that are in the cell membrane, right? By changing the pH, you might change how some of these proteins are folded. And if the proteins started to unravel themselves, to denature, then your membrane is going to start to have holes in it, and they will become more permeable as well. And lastly, the membrane composition. Okay? Don't assume all cell membranes are the same. Okay? I mean, they are similar in that they are made up of phospholipids and proteins and so on and so forth. But the percentage and the type of phospholipids that you find in cell membrane is not necessarily going to be the same for all plant cells, right? We talk about this in class actually. If you have a plant that is growing in a hot environment, there are adaptations that would make the cell membrane more rigid. And if you find a plant that grows in a cold environment, then you would have adaptation that would make the plant cell membrane to be more fluid, right? So you might want to review those as well. But the key thing here to know is the type of membrane that you have is definitely going to have an effect on its permeability. Um, so we are not going to be able to test all these things in our experiment. We are only going to be testing the effects of temperature on membrane permeability.